Hello everyone. Today we are going to discuss a new topic, that is sexual reproduction in flowering plants. Today in this video I will deal with the first topic in this subtopic in this, that is microsporogenesis. All of you must be amazed by observing diversity in flowering plants. You must have observed different colors, or you can say rich colors, different features in the flowers. I must tell you that all these features are because of a special type of, or you can say mode of reproduction, that is sexual reproduction. And we know that in number of organisms, this is the only type of reproduction which brings variation in them. In this topic, we are going to discuss about the sexual reproduction in angiospermic plants. Okay, before moving on, I just want to give you a recall that a plant contains two types of parts, one vegetative and the other one reproductive. We are familiar with this, that stem, leaves and roots are vegetative parts, whereas flowers are reproductive part. Okay, now we know that a flower consists of a sepal, petal, stamen as well as carpel. There are other terms for it like sepal, petal, they are calyx and corolla. Then again stamen is also called as androecium, and carpel or pistil is also known as gynoecium. A flower is a modified shoot which is formed after various types of structural as well as hormonal changes. Okay, all of them, that is sepal, petal and carpels, are appendages which are attached on a particular axis that is called thalamus. Okay, so before moving on, we know that flowers can be either bisexual or unisexual. Okay, that means they are having male reproductive parts as well as female reproductive part. So in this we are going to study about male reproductive part. In various types of flowers, you must have observed structures protruding out from the petals, and these specialized structures which resembles, and I've shown it in the picture, this is stamen. Okay, these stamens have got a general structure in them. That is, if you will look at them or observe them very carefully, you will find a structure, that is, every stamen consists of lobes. These are two lobes, as well as a slender stalk-like structure which is called filament. Okay, so anther is bilobed and there is a thin filament attached to it, and together it is called androecium or stamen. If you will cut a TS of the structure, okay, if you will cut a TS of the structure, you will find structure like this, where you can see two lobes. Okay, where you can see two lobes. This is first lobe and this one is the second lobe. Okay, in the first lobe you can see clearly that there are two compartments in each lobe. If you will look at this structures closely, you will find one and two compartments. Each compartment is called theca. Therefore, it is called as dithecous condition. Di means two, theca compartment. So two lobes having four compartments. Okay, each compartment you can see is empty, and young anther contains a tissue inside it which is called sporogenous tissue, and this sac-like structure which contains this sporogenous tissue is called microsporangium. This is called microsporangium. Okay, and when anther gets mature, this sac is called as pollen sac, as it contains pollen grains. So on the maturity, the anthers dehisces, or you can say ruptures, and releases pollen grains. Okay, and which is called as, you know that, and after that another process starts which is called pollination. So that is the other thing. And moving on, you can see here very clearly that this this is the TS of anther, where you can see a connective here. This is a sterile structure which does not participate in reproduction. Okay, and each anther contains theca, which contains a specialized tissue called as sporogenous tissue. Okay, and this is a structure of microsporangium. If we will see this microsporangium, you will find that each microsporangium has got four wall layers. Outermost is called epidermis, which functions in protection. Inner to it is endothecium, which helps in again protection, and inner to it are two to three layers are present, which are called middle layers. So there are three continuous layers: epidermis, endothecium, and middle layers. Okay, and innermost is the most important one, which is called tapetum. Okay, why we call it as most important? Because this will help in nourishment of developing pollen grains. Pollen grains are, I have already told you that pollen grains are present inside this. Okay, and these developing pollen grains will get nourishment or food from this tissue, which is tapetum. Now this is very important to know that what are the characteristics of this tapetum. Now tapetum contains, the cells of tapetum contains dense cytoplasm, number one. Second characteristic is they are multinucleate, having many nuclei. Function I have already discussed, that it helps in developing pollen grains. Okay, so this is the structure, this one is the line diagram, and this one is the cellular diagram. Okay, now moving on. Now before moving on, again I would like to tell you that young anthers contains this tissue, which is called sporogenous tissue. Okay, if I am saying tissue, it means group of cells. Each cell in a sporogenous tissue has potential to give rise to male gametophyte, that is pollen grain. Okay, so when we talk about sporogenous tissue, this each cell in the sporogenous tissue is called as PMC, pollen mother cell, or we can also call them as MMC, that is microspore mother cell. Okay, I'm repeating it once again. Each cell of sporogenous tissue is either called pollen mother cell PMC or microspore mother cell MMC. Okay, and later on this will undergo meiosis and it will form microspore tetrad. Okay, so that means PMC is diploid. After meiosis, it will form microspore, which will be haploid. Okay, now moving on, you can see this is a quick revision. Stamen consists of filament and anther. Anther is mostly bilobed, and anther is also diathecous. Okay, structure of microsporangium contains four wall layers: epidermis, endothecium, middle layers, and the most important one is tapetum. Okay, now moving on, you can see this is a small flowchart so that you can understand or remember the stages. Now we have discussed about sporogenous tissue. Each cell in sporogenous tissue contains microspore mother cell, is called as microspore mother cell, or MMC. It is also known as PMC. Okay, PMC here, it is also known as pollen, okay, mother cell. Okay, now this pollen mother cell is diploid in nature. It undergoes meiosis and forms microspore tetrad. Okay, this microspore tetrad is yet not a mature male gametophyte. It will further undergo certain changes to form male gametophyte or pollen grains. Okay, so we can summarize this process into a small term that is called as microsporogenesis. Okay, so microsporogenesis is the process in which microspores are formed from microspore mother cell by the process of meiosis. Okay, and these microspore mother cell will now undergo certain changes to form male gametophyte, that is pollen grains. Okay, now moving on, you must have seen anther and you must have touched them, and if you will touch them you will see that some yellowish powder will stick to your finger surface. This yellowish powder, if you will put it on slide on a drop of water and you will observe it under microscope, you will find number of sculpted structures like this. These beautiful structures are nothing but pollen grains, and these structures differ from one plant to another. Okay, now it is very important to understand about the development of this pollen grain. Okay, now in the first diagram here you can clearly see this one depicts or explains microsporogenesis. Okay, where microspore mother cell is diploid, it undergoes meiosis and forms microspore tetrad. That is, tetrad is a group of 4 cells. Here you can see 1 2 3 4 cells. Each cell is haploid now after meiosis, and this process is called microsporogenesis. Okay, now each microspore in this tetrad will start or undergo this developmental stages. Okay, here you can see that this is the microspore. It becomes, after some time, it becomes highly vacuolated and forms, and it contains number of vacuoles along with nucleus and cytoplasm, dense cytoplasm. Okay, the nucleus undergoes mitosis. Now meiosis will not take place, mitosis will take place. Okay, so now mitosis will take place in this nucleus, and it will, for a short period of time, we can see asymmetrical spindle formation here. Okay, and as soon as mitosis is complete, it divides into two cells. Bigger one is called vegetative cell and smaller one is called generative cell. Okay, and when a microspore contains two cells, that is vegetative cell and smaller one generative cell, then this is called two-celled stage. Okay, is it clear? Okay. And many pollen grains are actually shed at this stage, which is called two-celled stage. So you can say approximately 60% of angiospermic plants shed pollen grains at two-celled stage. So always remember and keep this in your mind that two-celled stage refers to one vegetative cell and the second one will be generative cell. Okay. Now before moving on, it is very important to understand the structure of pollen grain. Okay, there are two views given. One is the surface view, outermost view, and the other one is sectional view. Each pollen grain, mature pollen grain, consists of outer wall, that is exine, which is not smooth always. Okay, it is thick and it consists of a chemical which is called sporopollenin. Okay, along with it, at some places this exine is absent and that place is called germ pore. Okay, we call it germ pore because after pollination, when pollen tube formation starts, pollen tube formation cannot start from any part of this exine because it is very hard to uh, to break. So there are small pores already formed in it which gives rise to pollen tube, and these pores are called germ pores. Okay, now here you can see this is germ pore and this is exine. Always remember, exine is made up of a chemical that is sporopollenin, and it is very important, and it makes it resistant against high temperature, strong acids, alkali. And it is also observed that even enzymes cannot degrade sporopollenin. So that because of this reason, we have found fossilized pollen grains. Just imagine how pollen grains can remain for such a long period of time in the same shape because of the presence of sporopollenin, which makes it resistant. Okay, now moving on to another layer, that is intine. Now intine is inner to exine. It is not having any germ pore. It is thin, and it is made up of cellulose and pectin. So there is one difference. Exine is made up of sporopollenin. Intine is made up of cellulose and pectin. Exine is thick, intine is thin. Okay. Now this is all about the structure of a pollen grain. Now moving on, we have another diagram. You can clearly see that each cell contains you can see that pollen, this is a structure of pollen grain, where you can see outermost structure, outer wall is called exine, inner wall is called intine, and it contains a vegetative cell which is larger, and generative cell is here having generative nucleus which is floating inside it. There is germ pore which will help in formation of pollen tube. Okay, and because this vegetative nucleus is also known as tube nucleus, as it helps in formation of pollen tube during germination of pollen grains. Okay, so this is a very important part. Vegetative nucleus of vegetative cell is also known as tube nucleus, as it helps in formation of pollen tube. Okay, now I would like to explain one more thing with the help of this flowchart about two-celled stage and three-celled stage. Always remember that two-celled stage clearly means having one generative cell and one vegetative cell. This is the two-celled stage. But what do you mean by three-celled stage then? In some plants, generative cell, before shedding, generative cell divides into two male gametes. So, one, two, two male gametes and one vegetative cell. Okay, two, and this one is one. So 2 plus 1 becomes 3. Clear? So 2, that simply means that 2 male gametes plus 1 vegetative cell becomes 3. So this is called as three-celled stage. So it should be clear in your mind that if some plants shed pollen grains in two-celled stage, it simply means having one generative cell and vegetative cell. And if it is shed in three-celled stage, it means two male gametes with one vegetative cell, three-celled stage. Okay. If you will see the structure of pollen grains, that is when they germinate, they give rise to a pollen tube, and this pollen tube contains two male gamete, and here you can see tube nucleus, and here you can see this is the pollen grain forming a pollen tube. This is a pollen tube. Okay, and this pollen tube comes out through germ pore. Okay, and these male gametes are later on released in the embryo sac for fertilization. Okay, and this one is a microscopic diagram which is clearly showing the germination of pollen. These long tubes are nothing but pollen tubes, and these red structures are pollen grains. Okay, so I hope it is clear. Thank you.